Caroline. Can you give me the heads up when that's appeared your end, please?
Yeah, that's showing on my screen, and we're
Excellent
recording
job.
now.
Thank you. Morning, everybody. Thank you, Caroline, for inviting me again to speak today. A little bit of a background. I'm sort of new down here in the southeast, but I've been a tree health officer for nearly 8 years now. I've also been involved in tree and plant health since about 2002, so quite a long association with sort of looking at pests and diseases and all sorts of things, from sort of weird and wonderful things like magnolias and camellias through to sort of more sort of tree forestry based species that we'll be talking about today, like spruce and Douglas fir and things. So I'm going to, unlike yesterday, for those of you who were here, it was a sweet chestnut focus. Today we're going to look at 3 different hosts and 4 different things across those 3 different hosts. So we're going to look at Phytophthora ramorum and Ips cembrae and Larch. Ips, as we've already been discussing, there's Ips typographus that Nick's going to talk to you about in a minute, but this is a Larch specific species of Ips. We'll then cover a little bit about Douglas fir needle midge, and also pine processionary moth, which is in the same genus as oak processionary moth. So we'll get into ramorum. So again, for those of you yesterday, this will be a bit familiar. I thought I'd just take a moment to try and describe to you where Phytophoras sit with regards to sort of evolutionary branches within the phylogenetic tree. So over on the right hand side, you can see we've got animals, so us sitting in here, fungi, so that's sort of the true fungi where you're thinking about mushrooms and toadstools, over through to plants and green algae, and then you've got this unusually named heteroconts group. So Phytophoras, which are onomycetes, sit in this group. So these are water molds, so they're very different, as you can see, evolutionary terms, from fungi. They don't produce fruiting bodies in the same sense that you would expect from mushrooms and things. They produce spores, and we'll look a little bit about that in a moment. So, like all good things with genomics, we've really begun to understand Phytophthora. So Phytophthora is a genus. Before the marvels of gene sciences, we were looking at a few, well, I don't know, in the tens. You could count about 40 or 50 different species. We're now over 200 different species of Phytophoras. There are a number of exceptionally effective plant pathogens in that whole genus. As I said, we're going to look at ramorum for this bit, but in that same clade 8, there's some other interesting pathogens like Phytophthora lateralis and ostracedri and syringae, and some that you may be able to see as well in here over looking at Citricola as well. So there's a number of different really interesting pathogens, and some of you with really sharp eyes will see infestans, which is potato blight, sat all the way over there in clade 1. So all sorts sits within there. They're rather good at killing plants, unfortunately. But bringing it back into ramorum and larch as well, we'll sort of look at some of the structures that we find within ramorum. So they contain, or they produce, sorry, 2 types of methods of reproducing. So it's asexual reproduction with ramorum, homophallic. They produce sporangia, and on the end of there you see these sporangiums. Within there are zoospores, so they're tiny little, highly motile little things that have flagellums, tails, that you can see here on this slide. So they're able to move themselves around in relative scale, quite large distances. We also then have chlamydospores. So these are the resting structures. They can persist in the soil for at least 10 years that we know of and remain viable. So once they come into contact with a host, they can start the infection cycle again. Now these can either directly reinfect, or they can produce sporangia and then zoospores again. So a number of different ways of reproducing itself. These are rather ephemeral and will only last a matter of days, maybe sort of 10 days to a couple of weeks tops. We're saying these are the resting structures, and that's its survival mechanism. So trying to put into scale of what these things are actually like when we look at a leaf and looking at them in context with their host, you can see here all the tiny little sporangia on the edge of a Californian bay laurel leaf sample. So you can see them all growing out there. So they're really quite small in size. You know, not possible to see with naked eye. And then if you just squint slightly, you can see some swimming zoospores in here as well. So again, sort of just demonstrating the fact they're quite mobile, especially with the right climatic conditions. So Phytophthora need certain levels of moisture and humidity to survive, as I was saying, they're water molds, so they like humid and damp environments. So bringing this into a bit more context as to what this is actually like when you add the host into the life cycle. So here we can see we've got the chlamydospore on the outside of this diagram, where it'll emerge, and it can then either directly reinfect a host, or after, like I say, resting for a period of time, it can produce sporangiums that then go on to produce the zoospores, which go on to reinfect the host. These sporangium can also sometimes directly infect a host through hyphal penetration as well. So they rather adapt at what they do. Again, sort of thinking about why they're such a good plant pathogen. And so that just cycle goes on and on. It's affected by temperature and humidity levels as well. So more active in the autumn than it would be at other times of the year. Certainly within sort of when we start to think about larch trees, it's very much an autumnal cycling disease. Rhododendron is capable of cycling year round as long as the temperatures are suitable. But focusing back in again on larch now, and we'll start to look at some symptoms within larch trees of Phytophthora ramorum. So it's aerially transmitted, so we're going to start to see symptoms expressed within the crowns of trees. We're not going to be looking at the roots and things for this. It's up in the canopy. So the picture here you can see on the left hand side, larch tree out in the open, so shouldn't be showing any signs of unusual dieback, but you might be able to see those that aren't red-green colourblind. Sorry for those of you that are. You can start to see this foliage that is flushed in the spring, so it's come out flushed, it was okay, and then has suddenly died and turned that gingery colour. So this is what we term the sort of retained ginger foliage, because there's been a sudden change there. It's become infected and it's died suddenly, and it actually stays on that branch, sometimes through to into late autumn as well. It can hold on quite well because that sort of abscission layer hasn't formed because it's died suddenly. So that's one nice sort of symptom that you're looking at from a distance. Again, on the right hand side here, looking at a tree out in the open, we've got a dead branch here. So we're thinking, well, why is that dead? Because there shouldn't really be any signs. There's no sort of mechanical damage that we can see here. Then sort of thinking about water and transmission down, as we start to look at the branch below, we can see there's some foliage that's flushed in the spring there. So this might have died previous autumn, but this is flushed in the spring, and you can see it's a little bit of ginger foliage that's still retained there, and that's starting to work down as another point of infection, and on those branches below as well. So we've now got, from that single point, a number of multiple infections within that crown. And it's also sort of good to remember about that water droplet and water transmission as we start to look at some of the other slides as we go forward, looking at symptoms in rhodie in a moment. So now, again, sort of thinking of getting your eyes in for symptoms at a stand level, it's important to note the date on this one. So we're looking right smack in the middle of June, so everything should be nice and green. All those canopies should be green, should have foliage in them. And as you can see, looking through, it actually looks more like sort of October time. There's a number of crowns there that are either partly or completely defoliated. You can see some epicormic growth going on some of these as well. So here you can see sort of retain some of those sort of new, fresher epicormic shoots on some of these in the background, all indicating that something's obviously changed suddenly within here, because you've had chance for, or a relatively short amount of chance for epicormic growth to react to that. And then in the foreground here, you can see this rhododendron bush that's looking rather sparse as well. So again, all these things are starting to add up and making you think, what's going on this site? Why is it doing that? Then as we start to get a bit closer in on those that are showing defoliation and dieback, you start to see this resinosis. So what's going on here is that the disease has managed to work its way through those small, fine outer branch infections, worked its way through the larger scaffold branches and into the main stem in this instance. And the tree is reacting to this attack. The only way it can do that is by pumping a large amount of resin at it to try and get rid of it. So the resin contains all sorts of phenolic compounds, all sorts of really nasty stuff for plant pathogens that it's trying to push in there to try and kill off and rebuff this attack, but it's not working, unfortunately. But so it looks from a distance a bit like, again, like squirrel damage almost, but without any of that bark stripping, because you have all this resin bleed that then sets and goes this white colour. The slightly more difficult ones to see are the fresh ones, because this resin will still be clear, so you won't see them quite so easy. But again, you can start to see, if you look a bit closer here, it's a slight bit of epicormic reaction that's then suddenly died off as well, all indicating that this has been rather a sudden and rapid onset of disease within the tree. So now we're going to have a look at what's going on underneath the bark. So here you can see in the top picture, you've got some live healthy wood in underneath the chisel here. You've got some reactionary zones, so they go these crimson sort of colour. You can see it a bit clearer on that picture below. Again, this is the tree's defense mechanism in effect, so it's trying to put in all sorts of nasty inhibitors to prevent that pathogen getting any further through it. So not very good for sampling, because you actually don't get anything out of that of any worth, but actually as far as diagnostics go, it's really a useful feature, because it's not something we tend to see underneath the bark of larch for other things. And then as you start to work your way back down, you can see this necrotic brown material here. So that's where the disease has already been through and taken what it needs from the plant, from the cell structure of its host, and then moved on. So basically what the ramorum is doing is it's going through, destroying the cell structure, taking all of the starches and bits and pieces it needs to survive, and then moving on to the next cell, destroying that one and moving on and so forth. So it's, say, really quite an effective pathogen. And again, you can see a sort of a few bits of epicormic growth that have tried to flash out and then died off as well. This next slide was quite interesting. I collected these pictures just last year on a site up in North Yorkshire. All of this was, well, the rhododendron that you can see in the bottom right hand corner, which is really nice and symptomatic. You've got the sort of necrosis going on here, watery lesions running down through the midrib. That's about as pinned on classic symptoms for ramorum as you can get in rhododendron. This bush was sat just underneath this larch tree that was almost dead, looking like the infection had initially come down through this branch here, as well as from the top down. And as you can see, looking at above there, you've got all the white resinosis, little bit of epicormics that you can see on the floor next to the tree. And then you've got this necrotic material underneath the bark here. So it really is just underneath the bark in the cambial layer and that first little bit of sapwood, because that's where all the good stuff, as far as the pathogen's concerned, that's where all the good stuff is. That's what it's after. It doesn't go into the wood itself. It's just working through that cambial layer. And then below that branch there, again, you can see those nice crimson reactionary zones. And then top right, nice sort of picture here, clearly defined margin between that necrotic stuff and that sort of leading edge of the lesion, and then that really bright crimson reactionary zone. Nice diagnostic feature that I try and like to find when I'm going out sampling for ramorum. Then, as the season progresses on into the autumn, and I was starting to say about needing the right sort of climatic conditions. So as it's warm enough, but that humidity starts to increase in the autumn, it then starts to kick in to want to sporulate on larch. And this is the sort of the symptoms we tend to see on the foliage of larch. So it's wilted brown, looks sort of that similar sort of colour to the lesions on the rhododendron leaves, and it goes sort of a bit. I don't know how to describe it. I'm saying a bit manky looking, really. It's not a very good technical term, I'm afraid, but it is what it looks like. And thinking about how effective a host larch is for ramorum, thinking about a rhododendron leaf blade that can support a few hundred spores. The larch needles can support a few thousand. You then times that up over the biomass of the crown of one mature larch tree, and then factor in the fact that it could be, some 50, 100 foot up in the air in the nice mist. That's why it's such an effective host at not only reproducing on there, but then also going on for onward transmission, thinking about misty days and blowing from one side of a valley to another, or perhaps over the hill into another valley adjacent. So it really is quite an effective host, because you've had that. Unfortunately, with these two, they've never met each other naturally in the wild, so they haven't had the host has not had chance to evolve a reaction, the defense to the pathogen. You haven't had that coevolution. So when you put two a pathogen and a host that have never met, you get some rather strange outcomes sometimes, and unfortunately, this is one of them. Now, I've sort of danced around the edges of rhododendron as we've been talking about this as well. It's one of the key points in the transmission cycle, looking at sort of movement of people and sort of tools and machinery. If something were to come in with some ramorum spores that are viable, it's easy to come into contact with something like rhododendron because it's at ground level, and then that can start off a disease cycle within the woodland that then creates enough spore pressure that it can get from the rhododendron in the lower canopy, the sort of ground floor stuff, up into the larch tree. So being able to observe symptoms and identify symptoms within rhododendron is key for stopping some of those earlier transmissions. So what it starts off with is it looks a little bit like this. The wilted first image you see there looks a little bit like, well, as you would expect to see looking out the window if you could at the moment, of some frost damage or some cold reaction that rhodie does. But this is the sort of first indications on this fresh growth that the ramorum may be present. Then as it starts to develop, you get these necrotic dark lesions in on the stem, and they can also then work down onto the leaf blade as well. Again, thinking about sort of water droplets coming onto the top of this one and working its way down through the stem and those leaf blades. They're rather undefined watery edged margins that have sort of, I've heard them described as a dagger shaped lesion sometimes as well. They have quite a sort of point to them. And again, here you can see on this one that's just come up, water droplets here working their way down through the stem and then also through the leaf petiole and into the midrib. So rather a nice sort of running theme. And as that develops, here you can see a picture of a bush that's probably had an infection for a couple of years, most likely started at ground level. You can see this is all dead, but there's no retained sort of leaves here. As we go up to the next level, we can see that there's infection and dieback here, substantial dieback, but that's retained the foliage this time. Now that retained foliage can be rather waxy in texture, so rather being crumbly and friable like you would expect a dead leaf to be quite often, these are more like wax-proof paper. And then as we've sort of moved on in the number of cycles here, you can see just on the edge of the picture that wilting and the start of fresh infection in that side of the bush as well. So quite a nice sort of story of the evolution of infection within that bush there. So thinking about sort of control measures and our surveys for them. So ramorum is a quarantine organism, and as such, it's subject to statutory control, and that's achieved through statutory plant health notices. There are also biosecurity measures attached to those as well. It is also a reportable organism, so if you suspect that you might have ramorum on site or you've seen some symptoms, if you can report that through Tree Alert, that'd be great. Nice quality pictures as well really help for those triaging of those reports. Last year, COVID, as for everybody, has caused us issues. Our aerial surveillance program normally is a crew of 3 plus a pilot. We're now down to a single observer plus a pilot. So as you can probably imagine, that the workload on that single crew member has increased significantly. They're now operating a camera, filling out spreadsheets and trying to navigate at the same time whilst looking out of an aircraft down through a lens of a camera. It's an interesting combination of jobs to do in one go. We've also suspended multi-day flights as well. So we've got some good coverage this year considering all of those factors, but it's been tricky. But something that we did observe last year was a significant increase in positive larch sites. So we'll have a little look now at sort of what's happened over the past couple of years and where we've ended up as the end of last season. So back in 2019, we had around about 130 target sites, 55 SPHNs, and they were all really for quite small areas, quite often down to sort of level of single tree infections quite often. And those were all within areas that we know were infected in previous seasons. And that's been the focus of a downward trend since about 2012. Last year we had a significant upturn, so we've had just over 450 target sites, significant increase in the number of notices that we've issued, and those notices have also in some instances been in new areas of infection like the Peat District, up into the Yorkshire Dales as well. So there's been new areas of infection, and sort of thinking about Peat District and Yorkshire Dales, and it'll start to make a bit more sense when we look at some of the weather patterns in a moment. What's going to come this year? Will it be more of the same? I'm not entirely sure. The weather tail end of autumn last year was conducive. It was mild and it was damp, but yeah, I don't know. We'll have to see as we start to get up and start doing our aerial surveillance once Larch starts to flush later on this year. So this graph I'm bringing up for you now, I'll pick out a couple of trends within private woodlands that we've inspected over the years. So this blue line that's just come up, that's picking out the sites that we've investigated in private woodlands by season. So as you can see, sort of I was saying, that downward trend since about 2012. There's been a couple of blips, but it's been generally downwards. And even this one, although there was more that we looked at, that was at a lower scale. So we're talking single tree infections. The other line that's just come up now is the number of notices that were served on those private woodlands as well. Sorry, just to go back on that one. So thinking about context for where we are from last season, for private woodlands, we're probably up to around about this point with regards to the number of sites that we've looked at, and probably somewhere around about here for the number of notices we've issued. So again, quite an upward tick. So why was that? As I was saying earlier on, it's looking at weather in the previous autumn that dictates the level of infection the following season. So we'll look at autumn averages for temperature and rainfall for GB, particularly focusing in on England though. So autumn 18, a little bit warmer than normal, but nothing significant. A little bit drier than normal for most areas and significantly drier for some. So 2018 was not that conducive. Autumn 2018 wasn't that conducive looking at in context of ramorum. So now let's think about temperatures and rainfall for autumn 19. Temperature a little bit cooler or about the same really, not a huge change, but when we start to look at the rainfall, significant change. So we've got a far higher than average rainfall in the Yorkshire Dales and the Peak District. So it's interesting. I'm not saying that's the only factor, but it's definitely a significant factor in why we've seen a shift in the number of sites that we picked up on last year. So we'll move into cembrae now. It's cembrae. So quite a wide distribution, found pretty much everywhere that you find larch within its native range. First record in Great Britain was in 55, primarily a secondary pest species. Now that sounds a bit like an oxymoron if I'm not careful, so I'll describe what that means. It likes to attack stressed and dying trees, sorry. So it doesn't instigate an attack, it will take advantage of a larch tree that is already under significant stress for one reason or another. We do observe it on larch that is infected with ramorum, and we have seen it in other instances as well where larch trees are significantly stressed. Quite often we tend to see a lot of woodpecker activity with cembrae as well, because when it can build up quite a decent sized population within the tree, so quite attractive to predator species. It tends to, well, it is, you find it in single larval gallery as well, which I'll show you what those look like a little bit in a minute, but you don't find it in a big mass like you would do with something like Dendroctonus. These are very clean and tidy as far as their gallery production. This is what it looks like. So for scale, that's probably about 4 mil long roughly, so not very big. To get a positive identification for what it is, you need to look at it underneath a microscope, because there are a few other bark beetle species that look quite similar. So I'm sure Nick will in a minute, when he starts talking about typographus, show you similar pictures, but this is in larch still, remembering, so we're looking at cembrae. Nice clean maternal galleries, and then you have all the larval galleries where there's a single egg in each of those galleries, so you'll have a single larva developing within each of those galleries off the side. Quite nice and neat. A little bit of a sort of case study of a site I looked at up in North Yorkshire a few years ago. Clear fell about 4.5–5 hectares, was quite sheltered on a free draining site, so quite sandy here. And you can probably see these larch trees just on the side here starting to show quite significant signs of dieback. So the forester was a bit concerned, gave me a call. I came out, had a look, and yeah, there was a lot of dieback in there. He was quite worried that it was ramorum. After felling one, I could see lots of signs of insect activity in here. You can just see individual little larval galleries off the side with this one. I went back the next day because I couldn't quite manage to finish up all my investigations, and it was that active that all of the frass, this is, you can see all this sawdust coming out here, that's all the activity from the beetles that are underneath the bark. So when it was stood up and felled, all this fell off. Went back the next morning and it was all covered in frass again, so really quite an active one going on there. Didn't manage to clarify whether that was primary or secondary. I think probably a secondary looking at the site, but it can really have quite an impact if you've got stressed larch trees on site for one reason or another. One of those reasons is quite often drought stress. So now we'll go into having a look a little bit about Douglas fir needle midge, found in North America and a few places, so France and Germany and Low Countries, not that widely distributed, not present within GB. This next slide I brought up for you is the risk register, which is one of the ways we use of prioritizing risk levels against pests and pathogens, those that are present and those that could be introduced, and looking at pathways and things. Relatively low risk rating on this one, really, when you consider it's 36 out of 125. So, but interesting all the same, because this would be one of those that if it did arrive, I think it would be a long time before we actually noticed the symptoms of it. So looking at life cycle, we'll start up in the top here. The mature larvae overwinter in the soil, they pupate and emerge then as adults in around about April, May time. May quickly go on there, have a lifespan of about a few weeks, so really quite short-lived things. But they'll go on to mate and then oviposit, so lay their eggs in the forming buds of Douglas fir for that year. So thinking about the fact that it's, you know, those bud bursts, it's the symptoms that you're going to observe are going to be in the current year's needles, because that's what it's attacking. So those eggs hatch and develop over a fairly short space of time, go on through a number of instars, and then start to cause damage within the needles. So they cause, as they develop, they sort of cause a gall, as the name suggests, on the Douglas fir. So looking at it from a distance, what you're likely to see is this sort of chlorotic current year's needles. So not, you know, doesn't look too dissimilar from a number of other things, really, from a distance at least. When you start to then work in a bit closer, you can see that, you know, this is an extreme example, but this one that's come up, you've got all the current year's growth extensions. They've all gone this sort of chlorotic yellowy colour, and they're not happy at all. Then as you start to look in closer, you can see flecks and bands on those needles. So here you can see sort of a few flecks and bands and some swelling a little bit as well. Start to look in a bit closer. Now you can really start to see what's going on. This bunch here, you've got a slight swelling of the needle, and you've got that sort of chlorosis either side. And then when you open up those needles with those galls, you can start to see a little larva in there that's causing that. So it's creating the plant is responding to the presence of that larva, and it creates an abnormal growth around it. That's what causes the gall. So now moving on to pine processionary moth, and those of you that are familiar with oak processionary moth will recognise the genus. So it sits within the same genus, so has a number of similar issues with regards to urticating hairs, which we'll talk about in a bit in a minute, but currently not present within GB. It is around the Mediterranean basin and through into North Africa, likes pine, as the name suggests, but also will go in extreme cases onto cedars as well in some instances. So can be, yeah, can have a couple of 3 different hosts that it likes. One of the other than the fact that it's on pine rather than oak, one of the other useful differences between oak processionary moth is the life cycle is skewed to the other side of the year. So with oak processionary moth, we're looking at a lot of summer activity. With pine processionary moth, we're looking at a lot of winter activity. So it lays eggs in sort of around about July through to September. These times are all approximate because they're obviously worked on the areas that they're in, and the climate here is slightly different. So the thresholds for temperature days will be slightly different. But anyway, so you'll have eggs appearing in sort of into the late summer, early autumn. They develop through a number of instars, a number of growths, and you'll get those develop nice silken nests up in the top of the tree. So a really good diagnostic feature for spotting these is these sort of large, can be football-sized nests up in the tree in the winter. Again, very useful from a import inspection side of things for limiting the risk. We move plants in the winter generally when they're under, causes less stress for them. So with regards to visual inspections, far easier to see than something compared to oak processionary moth, where if you move it in the winter, all you're looking for is a little egg plaque. So quite a different visual cue for inspections. So that carries on through the winter till about sort of February, March time, when they process down the tree into the soil. They pupate in the soil and then emerge as adults in through till about sort of June time. So caterpillars look slightly different from oak processionary moths. So these have got rather consistent hair pattern on them, other than sort of oak processionary moth is spiky and inconsistent. This is nice and sort of generally fluffy, nice sort of dark blue bands in between those orange hairs on a stripe along the back. Here we can see, so egg plaques are on needles rather than on the branches, so they would be quite difficult to spot. But luckily, as I was saying, at the times of year that we're moving plants from the continent, that we wouldn't be looking for plaques. We'd be looking for the nests. Here you can see a nice shot from above, that nice clear orange stripe down the back intersected with those bluey black bands. And this is what I'm talking about. It's nice and easy to spot. This is rather an extreme example, but you can see it's a very defoliated tree here. So again, all sort of thinking back to the Douglas fir needle midge and this one, they're both defoliators. So in their own right, perhaps not necessarily going to cause mortality, apart from in extreme cases. It's the fact that they weaken trees and make them susceptible to other things. And then also with pine processionary moth, obviously the public health issue with it as well. But nice, easy, big nests to be able to see when you're inspecting for stuff in the autumn and winter. And those are chrysalises that you'll find in the soil underneath the tree. And as those of you that are going to go onto the OPM talk later on this week, I would describe pine processionary moth in the same way as my colleague Andrew does. For oak processionary moth, it's an uninteresting little brown job. It really doesn't look that interesting as a moth in its own right. It's really for, unless you were targeted and looking for it, it's probably not one that you'd notice. And a couple of fun images here from the continent. Control for pine processionary moth used to be through spraying, but that's not possible now. So there's cultural practices now. They use this nice, interesting trap you can see on the left-hand side as taking advantage of that habit of it processing down the tree to pupate. They're catching the caterpillars in this bag here. So that's quite a neat control solution. And Andrew Hobbit very kindly sent me this picture of a processing pine processionary moth all the way around this bin. So I don't know whether somebody dumped a few nests in there and they've escaped or what. I'm not quite sure, but interesting to see that habit sort of continuing around a bin rather than down a tree. I shall finish up now with this slide just to leave you with a few links and bits and pieces and other resources, and just to say that if you are spotting things that you're not sure about, please do report it through Tree Alert. But the one thing I would ask is make sure you supply some decent pictures and some decent location details so I can follow that up. And thank you very much. I shall pass back to Caroline.
That's lovely. Thanks very much for that, Alan. No one's posted any questions. I know we're holding questions to the end anyway, but people may want to have a think and see if there's anything they want to ask about that. But if there's, with no further ado, I'm going to hand over to Nick Hazlitt to talk about Ips typographus, depending on how you want to pronounce it.
Right. Thank you, Caroline. I'm just looking to share my screen and share the right screen. Bear with me 1 minute. Are you, Caroline, can you see that?
Yep, I can see your general PowerPoint. Yeah.
You can see the okay. There we are. Good morning, everyone, and thanks, Alan. That was very interesting. I'm Nick Hazlitt. I'm the regional manager for the plant health team for Southern England and Wales. I spent the 90s as a forestry contractor in establishment survey work, particularly. I joined the commission in 2002 and spent 12 or 13 years as a beet forester, and then I joined the plant health team in 2014, I think, just when lots of things were kicking off. A lot of my work is focused at the border, and I manage the inspections of timber, wood products and wood packaging that are being imported through many of our ports, in an effort to ensure we're not bringing more pests and diseases in through the trade pathways. I've also more recently got involved in some of the outbreak management work, which brings us to Ips typographus. And I'm going to try and give an overview of a bit of background to the pest, the control, and touch on surveillance and the eradication work we've been dealing with for the last couple of years. So this is really a catch-all. A lot of these points tend to blend into each other. I'm trying. It's a huge subject, and I'm trying to pick out the very salient points, both in terms of the background and then our actions. So, what is it? It's an insect. It's in the order of Coleoptera. There are the subfamily is Scolytinae and the genus is Ips. I think there are 37 Ips species, all bark beetles, in the world, mostly found in the northern hemisphere. This particular species, Ips typographus, is endemic through much of Europe and Asia. It is one of the three. There are three Ips species present in GB, but this is not one of them. Just touching on the point Caroline made, the beetle is known by a whole host of names: larger eight-tooth European spruce bark beetle, the spruce bark beetle, eight-tooth bark beetle, European spruce bark beetle, and so on. I like the engraver name because that says that describes it very well. There are also, I think, 6 common names that the Germans use and 4 common names that the French, or 5 that the French use. So helpfully, it has its own code, the EPO code, so that we all know exactly what we're talking about, and is more widely known as by the scientific name Ips typographus. But there are also 2 other recognized scientific descriptions of this beetle. It's a bark beetle. There it is, as often described, as Alan mentioned. It's a little brown jobby. It's about up to 6 or 8 centimeters long. There are a number of other bark beetles that look very similar. It gains its name from the fact that it's got these 7 points here, which are described as teeth at the back of the clavicle. We have 3 species of Ips established in the UK, and they are they're present either in part or most of UK, and are generally bumbling along without too much impact. There's surveillance and consideration for how behaviour might change in respect to changing climate and changing conditions, particularly around Ips cembrae. But we are aware of these beetles, and there's ongoing surveillance for them. Ips typographus has a very high risk on our risk register, and you see that the likelihood, the impact is all 5 with unmitigated risks. So it is of great concern to the sector. It is a devastating pest in its current natural range. Fortunately, there are some potential climatic limitations for it, but as we will see, we've had first-hand experience of this this pest. So, as a brief biology, it's generally a secondary pest. It's a keystone species in the natural environment, so it's really part of that cycle of decay and succession, and many of the larger natural forests in Europe can be, particularly the spruce forests, can be traced back to significant events over the last 300 or 400 years, where bark beetles have in effect reset the succession. Although it's a secondary pest, in the right circumstance, it can develop into really a primary pest, because the competition for resource builds up to such a point that the beetles start attacking very healthy trees. And I would say that generally it's a secondary pest which will inhabit trees that have got weakened defences, trees that have been weakened by wind, fire, drought, poor conditions, extreme climatic conditions, and their natural ability to rebuff the pest is reduced. A healthy tree will often be able to provide its own defence mechanisms by in effect pumping resin up into the galleries and actually stopping the beetle in its life cycle. But weakened trees, or trees that are overcome by huge numbers, don't have the resource to do that. The beetle lays eggs under the bark. As the larvae develop, they start feeding on the cambium layer, particularly between the bark and the sapwood, and can rapidly lead to death as a result of interrupting the natural process of food and nutrients for the tree. They can breed very successfully, very rapidly, with 2 or 3 breeding cycles in a year in good conditions. So once they've got a foothold, they can very rapidly spread. The males initiate an attack, and they follow plant volatiles from the host trees, releasing an aggregation pheromone which attracts others, both male and female, to the party in effect. So trees that are stressed by wind or harvesting or damage are releasing lots of volatiles which attract the insect and provides the opportunity. There is a delay in the development through the winter, when the insect will go into a diapause, when whatever stage of development it's in, it sort of goes into a stasis, either within the tree under the bark layer in the pupal chamber, or particularly into the leaf litter, or even into a second tree. My entomologist colleagues suggest it's a pretty lazy beetle, so won't often make efforts to go very far, or the majority of the population won't. So crawling down into a nice leaf litter layer is very appealing and very easy, and that has implications for how we manage and consider any infestations. The distribution, as I referred to, you can see this is from the EPO map, and it shows that typographus is known to be found right across Europe and much of Asia, with the I think this is Serbia and Macedonia, where it's very possible that it's present, but we don't have records. And this represents the outbreak that we discovered in GB in 2018. It's an indicative map that, which looks to be sitting over, I guess, the North Midlands. The general position with outbreaks is they last for a couple of years following a major event. There are beetles, small numbers of beetles in the general population pootling along. An event of fire, flood, stress of some sort then prompts a significant and very rapid rise in populations, and this has been going on for many years. It may be that the cycles are becoming slightly more frequent, and the numbers of trees affected and the volume of timber affected is quite staggering. This is data from the early to mid 90s, which shows that 30 million trees were lost in that outbreak across central and north Europe. We know in the last 4 or 5 years there's been very significant outbreaks in Germany, in Belgium, in France, which one has brought millions more beetles to the party or into the environment, but also brought them much closer to GB, which is significant in terms of our infestation. It has landscape scale, social, economic and ecological consequences. It's very rapidly colonizes right through a landscape, and both in its natural range and in plantations. Spruce, which is the primary host species for the beetle, is often found in monocultures or in significant plantations. So there is very easy dispersal for the beetle across large landscapes. And thank you, Alan. This is another photo of the little brown jobby. It's a 1/4 inch long, but has this devastating effect. Just a couple of indicators of its presence. It's very difficult from just a visual inspection to identify the presence of the beetle, because the indicators could be illustrating many other forms of decay. Once you've got your eye in, you can certainly generally pick up a nips beetle, and if you're very clever with a hand lens, you can identify its typographus. The galleries that it's producing, both under the bark, on the inside of the bark, and on the cambium layer, are very distinctive. So that is helpful when we know we've got an infestation and we're looking for the extent of it up on the ground, supporting other surveillance work. So we have, I guess, we've been keeping tabs and keeping an eye open for this beetle for many decades. It forms part of our protected zone status, which provided us with some protection in terms of potential host material being imported into GB, and is one of the reasons why the vast majority of conifer imports coming into GB were entirely bark-free square sawn material, because whilst the beetle is present and endemic throughout Europe, being not present in GB, we still have that level of protection for it. We also have had nearly 30 years of surveying for it through really through two methods. One is with a series of survey sites across the country, where conifer billets were baited with a pheromone lure, and the pheromone lure is aimed at attracting bark beetles, and at the end of the season, through the season, and then at the end of the season, these survey sites are dismantled, and a very close inspection of any of the insect activity is undertaken. And those surveys have been going on since 1993, predominantly in the major spruce growing areas, but in 2015 we extended the range of those to also increase the number of sites next to the major processors and railheads and so on. And we've never, until 2018, we never found a Ips typographus in any of these traps. In addition to those forest-based bark beetle survey traps, we also have pheromone-baited lures at ports and processors, and we've periodically over the last 20 years had occasional findings in these traps, which generally represent hitchhikers with imported material. In very low numbers at the ports, they're not really problematic. There's neither the host material nor the numbers significant enough to start a breeding population, but we're aware that we've been finding these periodically. Now, in 2018, we actually discovered in one of our baited forest billet traps, we found evidence of Ips typographus. Unusually, it was actually in a stand of pine and in pine billets. The infestation was confirmed in December 2018, and immediately, really, we started following the contingency plan, which straight away put a movement restriction in place for any material from that part of the country, and we had a very rapid response and take up from the principal forest operations and forest management sector. We had engagement with landowners around the outbreak site, and that was followed up by formal legal statutory plant health notices, which restricted further movement in that material. Then very quickly, the Ips typographus order was laid and went through Parliament, and that was followed by the notice that supports it. And I'll say a bit more about that legislation, because that really gives us the framework under which we have continued to control the outbreak and support the eradication program. So the decision process, as I said, was based on the contingency plan. That's published, it's a PDF, it's published at this link. Like a lot of contingency plans, it's a good plan as a starting point. Never having had to deal with an outbreak of Ips typographus before, that plan has been reviewed and is undergoing further revision at the moment, and there have been slight diversions from the original anticipated process, but very rapidly the Forestry Commission were given the role as a lead government department, and an incident management team set up. The incident management team delivers the operational aspect of the work, engaging with the landowners, delivering the operational eradication work, setting up the surveillance and monitoring, and it comprises of professionals from the tree health team, the plant health team, forest research. We've got comm support in there. We engage with the private sector, with the processing sector, with the landowning sector, with forest services. We're steered in terms of policy by the lead government department, but that's obviously a bit of a two-way conversation. The legislation that I referenced was a statutory instrument laid in 2019, and the overarching legislation which is the plant health typographus order, it allows us to demarcate areas around confirmed outbreak sites and imposes movement restrictions on conifer material capable of spreading the pest using a notice. So this is the overarching legislation under which we operate, and that's been laid in such a way that at the time, should we have determined any other outbreak sites, it would have given us some ease in terms of how we manage that. As it happens, we've only had one site of infestation, so that has simplified things. As it happens, we've been able to remove, or we've been able to specify spruce material in the notice, whereas if it came to it, we could put controls on all conifer movement within any outbreak or demarcated area. So in this order, we have the provision to enable plant health inspectors to authorise movement and processing of relevant material with bark from the demarcated area, where this can be achieved without risking the spread of Ips typographus. And this is an important factor both in terms of supporting the sector and particularly in terms of how we promote good biosecurity practices within that demarcated area. The legislation can be found through this link, and that links to some other bits of supporting information. So following a fairly rapid survey around the infested site, which, sorry, I didn't mean to go back then, which going in the wrong direction, I'm afraid. So there was rapid surveillance around that infested site to really delimit the area of infestation, the extent of the infestation, and that was through very intensive ground surveys immediately around the infestation site, going out to 500 meters, and then degrees of both survey, site-based inspection and other forms of survey going out from that point to a radius of 50 kilometers. I'll hopefully be able to have time to touch on that a little bit further later on. But that gave us the basis for the introduction of this demarcated zone, which was described in the notice number 1. The notice describes exactly what the relevant material is, and whilst the order that stands above this notice allows us to control any conifer material, we made this very explicit to Picea, because from our investigations it was apparent that the only time we found the beetle present in non-spruce material was when we'd invited it there with our pheromone baited lures. So it's explicit to the genus Picea. It's explicit to all wood, bark, wood waste, and so on. And this last point, 3.C, is important because it includes trees over 3 meters in height, which brings Christmas trees under this control, although not the usual domestic Christmas trees, cut Christmas trees, but rather the larger civic trees that are over 3 meters. And that's because trees under 3 meters are deemed to not be sufficient to host the material. They're certainly not large enough for any breeding populations to establish. So this is a demarcated area. In effect, obviously, we're nearly coming over to France here if we'd extend it to the full extent of a 50k surveillance around the infested site. But this allows us to and requires us to have control of any movement of spruce material within this area, which is both a precautionary and a statutory requirement for us. that brings us on to moving spruce, and there's a question of why would you move spruce? Why would we want to be in a position where we're moving material from this demarcated area, which could possibly host the beetle, but that might be actually moving it to uninfested parts of the country? And as an explanation of that, that's because firstly, it's an overall part of the strategy to help manage and maintain spruce health in spruce stands. It promotes good forest movement by allowing us to, or allowing landowners to take action on their woodlands without having to destroy the material within the demarcated area. It brings it to market, so it supports landowners, the harvesting sector and the processing sector. It gives us the mechanism to place controls and inspections at all stages of the process. And I would say that we're only allowing movement of material found not to be infested. There's a slight note on that in that we allowed movement of material from the infested site under very strict controls to a place of destruction, and that was material that was known to be infested and known to be potentially carrying beetles. So there was a very high degree of control in terms of how that material was moved, when it was moved and where it goes to. We've still maintained that level of control at a high level on any other movement of spruce from the demarcated area, but there are opportunities for landowners and the sector to move spruce, which promotes one, good forest health, and secondly, supports some revenue streams for the sector. The outline process for moving spruce is that a business would need to be a professional operator, and there is a process for applicants to become a professional operator, and they do that in conjunction with an online e-learning module. They're given outline approval to move spruce from within the demarcated area once they've got that, and then individual movements are controlled with a further level of inspection and regulation. Once a business is approved to move the material, they will provide a request for an inspection. Upon successful inspection by a plant health officer, they will be given a letter of authority to move that material, and they're then allowed to move that material to an approved site for processing or destruction. The processing or destruction must take place at the approved site, together with any residues arriving from that processing. The movement authorization really provides us with an early intention of the movement of material. It gives us an opportunity to reference other data that we have in terms of other survey work, flight surveys, data from pheromone lures that we have within the demarcated area. We will inspect the standing trees within the stand, and then either following or during harvesting, we'll give a close examination of produce at stump and in the stack to again verify that there isn't any evidence of the beetle in the stand. Now that is both a regulatory requirement, a legislative requirement for us to do that, and it's obviously very good practice, because however well we survey any woodland, there's only ever a guarantee of finding something, not confirming its absence. But there are multiple layers of inspection and consideration before we allow material to move from any site within the demarcated area, and the material then has to move to a processor that meets our requirements in terms of how quickly they process that material, the manner in which they process it, the manner in which they handle any of the co-product, and the traceability of that material through their record keeping. So this was in fact, it was a Christmas tree, an overstood Christmas tree plantation that's been felled and removed. It was a relatively small area, but was really very good condition for ips infestation, because as an overstood plantation, it was under-thinned. The trees were all stressed for a variety of reasons. I don't think it had been thinned since the day it was planted. So the trees were under stressed, with 3 warm exposed edges, which if there had been the opportunity, its beetles may well have settled there very rapidly. It's of benefit to everyone that stand is no longer there, but it was facilitated because there was some recovery for the landowner through the authorization process. So that's a very brief outline of the movement requirements. I touched on the fact that the material can only go to authorized processors, and there's similarly a process where we review and give authorization to processors. Any business can be authorized to receive and process spruce from a demarcated area if they can meet our criteria, and that business can be located anywhere. There's no restriction on where that process is, but the location of that process will give us, we will be considering the precise location of that in terms of the risk that brings, both in moving the material and then what happens to the material when it gets there. We need processors to be able to demonstrate that they have the appropriate biosecurity measures, understanding and record keeping in process, so that we can trace back material. We need them to go through an application process, which allows us to have that engagement with them, and to ensure that everyone's aware of the risks and responsibilities. We will then inspect the premises and give an approval for that. Now, this is quite important in terms of both how we manage the infestation, how we manage the ongoing control of movement of material, but it also has some impact in terms of the readiness for landowners to move material to market, because there is an element of additional bureaucracy and there's some inevitable controls that we have at the processors, the processor sites, in order to remove any risk of us transporting and spreading that beetle through in numbers that would likely lead to any breeding populations. So we have had limited number of processors at the moment who are authorised to take this material. The primary processor is the KRE chip power plant at Sandwich, and that's a very good site in from many perspectives. It's actually within the demarcated area. It's about as far east as you can go, so it's actually taking any material away from the edge of the demarcated area. The material is all ends up going up the chimney. It's located in an area where there's very little host material in close proximity to it. It's a very modern setup, so there is very good control of material movement in and out of the site. And we have the option of taking material there that has been chipped on site, which is the safest approach, or taking material there in the round, which is also helpful, certainly in theory, in terms of moving unmanaged stands on. However, there are some difficulties that are encountered with a plant that's operating 24/7, which has a continuous flow of material, that has huge stocks of material there, and the volumes of spruce that we're currently moving around in the demarcated are quite low. So it's quite a challenge for the power plant to meet our requirements in terms of how quickly they handle material, what they do with the co-product and the arising. This is the sort of sweepings they have from the log yard and from the loading conveyor that takes the roundwood into their chip hall where it's chipped. And it's quite onerous on the business to stop and sweep down and clean down every time they have a small volume of round material to shift. It's a much easier process for us if they're taking material that's chipped on site, because that moves in covered wagons, walking floor trailers, is tipped straight into the fuel hall, the chip hall, and goes in effect nowhere else until it goes up the chimney, until it goes into the burners and is incinerated. The downside of that is that it's very costly to chip material on site. We have to do that where there is any infestation, which currently has just been one site, and that's the hope. But with small volumes of material around the demarcated area, it's quite a it's quite a financial burden to chip on site because of the cost of taking machinery into the site that will do that. Christmas trees, as I mentioned, are also subject to our inspections and our movement controls. We've had engagement with the British Christmas Tree Growers Association. In effect, there's a similar process for Christmas tree growers wishing to move Christmas trees either within the demarcated area or outside of it from within the demarcated area. There's a similar inspection process of the Christmas tree stands. There is no requirement for recipients to be authorised processors, but we do ask that suppliers provide guidance and instructions to their customers that would prompt chipping or burning of a Christmas tree at the end of the season. Christmas trees are by definition or nature generally very well managed, very heavily inspected. There is very little opportunity for establishment, both in terms of the condition of the trees, which isn't attractive to typographus. They're monitored and surveyed and often treated with insecticide fairly readily. So healthy and in cycle Christmas tree plantations are of very low risk, but overstood Christmas tree plantations that become stressed and in poor health are a different challenge for us. This information, it's the map on the left here shows the two authorised processors, current authorised processors. There are a couple of others we're talking about, but principally this is the power station in Sandwich, which takes in roundwood of virtually any size or shape, and Thompson's sawmill up here, which is particularly keen on conifer log material. And it's very helpful that Thompson's have come on board with this process, because it's a very valuable and important market for the sector, and will allow a little better return than sending everything to KRE. Thompson's, the mill here can really meet our requirements because they have a very rapid turnover of logs coming in. So we know that any logs going up to site won't spend any time there. They can be processed in relatively small volumes, and because they've got very close control on all their co-product, because that can be utilised on site in the biomass boiler they have there. So it's a sort of end-to-end control we have. Any material entering that site we know will either leave the site entirely bark-free as sawn and possibly treated material, and any co-product will be burnt on in the biomass boiler there. This was the number of inspections that we've done really up to 14 months ago, I think, and we've had a similar level of inspection carried out this year across spruce stands throughout the area. This is just reference to the application process for authorised processors. They can go online and pull down the application process, which then prompts an inspection, as I see, and then they're provided with this authorisation material for traceability, the records of dispatch from any site with the weight, the time, the site that it's moving from, and its destination are recorded. So at any one time we can follow up any one dispatch to any of these processors, and that's a sort of audit process we've checked on a couple of times to ensure that it's working as designed. So that's really an outline of the control process for it. I've got a few minutes, so I'm going to run into very quickly just into a little bit of discussion about the eradication process. I referenced earlier that the immediate response to determining the infestation, this is an aerial view of it, was to and that's that's a site, was to determine the extent of opportunity for the beetle in the immediate environs. So there was a series of which are ongoing surveillance surveys of one way and another. Everything on the main infested zone, the materials were felled, chipped on site, all the brush was burnt on site, all the stumps were lowered and burnt on site, and there was an endeavour to rake up and burn all the litter layer, because as I mentioned, the insect can overwinter in the litter layer in huge numbers. At a 300-meter radius from the infestation, everything was removed except small healthy trees, spruce we're talking about, and a slightly wider survey then determined anything that was heavily stressed, dying, or decayed was also destroyed. And then there's an ongoing survey program which is recording, looking and surveying spruce, initially up to the extent of the 50k demarcated zone, but then a much wider ongoing program through different surveillance techniques. In addition to the initial felling and burning of any potentially infested material, there is an ongoing trapping program which is utilising pheromone-baited traps at very close intensity across the infestation site, which has been hoovering up any insects that remained in that litter layer. This is two esteemed entomologists, Nigel and Nick. Max Blake didn't manage to get himself in this photo because he's taking it, but Max has been very integral to the whole eradication and ongoing surveillance work. This is just a few photos of the initial rake and burn. Very good response from the landowner, who was very committed to really doing whatever is necessary. It's a costly operation for any landowner because the cost of this operation falls to the landowner. It's not supported through direct funding, certainly, and it's also quite a significant impact on a site like this to have harvesters, forwarders, significant volume of rake and burn. So we were very pleased to receive the support of the landowner in this, both in practical terms and their commitment to help the eradication process. very important to get the brash burnt and the litter layer. Slightly contrary to lots of current practice in terms of how we're managing stands, particularly in pause areas and on multi-species sites. But the importance of good forest hygiene was very important. Was critical here, and this was the material from the infested site awaiting chipping and dispatch to KRE. And this again did allow some return to the landowner to support the very costly operations. The ongoing eradication and control is really through three methods. So we've got this very intensive pheromone trap grid across the site. That's completed 2 years of trapping, and we have hoovered up very significant numbers of beetles that remained across the site in the litter layer. The first year we also retained some standing trap trees and implemented some trap log piles. That was part of the eradication, but part of our ongoing understanding of the beetle and how it moved. There is additional monitoring right throughout the demarcated area and to a much further wider environment now. The traps are emptied twice weekly through the season, and all those beetles, as a quarantine beetle, are then sent to Alice Holt for counting and for disposal. This is the pheromone. This is a picture of the pheromone lure, which is a funnel trap baited with the pheromone that sits in here. The beetles fly in, hoping for a party, drop down here and are captured in a glycol solution here, which is checked and emptied twice monthly on a fortnightly cycle. The wider network of pheromone traps is indicated here. This is again from a couple of years ago, and there are additional traps supporting this program, both in terms of surveillance and control, which are illustrated here. So all these diamonds show where we have the ongoing forest-based pheromone-baited beetle billet piles, which are surveyed annually. Again, a similar process. The pheromone lure is added to the freshly cut billets and then is visited 3 times during the season before a destructive sampling at the end of the year when all the bark is removed. It was through one of these existing pheromone billets that we first identified the outbreak site. And since the outbreak, we've increased the number of and intensity of sites to include some of the wider environment, and increased the number of pheromone baited lures that we have at ports, piers, and processors, which are potential pathways for the beetle. As further research and background, there's a lot of work going on into the suitability of other species, particularly Sitka spruce, obviously, and using billets to assess the survival and growth. There is work predicting the source and founder population size. We're looking at genome sequencing and looking to try and determine whether we can gain any insight into where these populations have come from. The understanding is that these beetles have been blowing in from the continent for the last couple of years, prompted by very high numbers on the continent, supported by extreme conditions on the continent with very long summers, very extended breeding periods, and then have found themselves arriving in the hot and sunny southeast of England in the summer of 2018, 2019, which rather than the usual cold, wet August that they may be faced with, and have come over in such numbers that they've managed to get a foothold and then probably carry on breeding for a couple of years. It's helpful if we can understand exactly where they've come from, but that's a very difficult thing to determine exactly. One of the other important elements of Ips typographus is it's a vector for blue stains and other pathogens, and there's work going on to understand that that pattern and that characterization by the Ento team at Alice Holt, and further work trying to understand population dynamics. We're fairly confident that you need at least 10 beetles arriving at the right place in the right time with an appropriate host to have any chance of forming a population, but there's a lot of work to understand exactly how that works in behaviour in GB. There is certainly an awful lot of information I could provide on either the behaviour or on the survey work or on the detail of the eradication programme, but I really wanted people to understand the legislative framework we're working in and the controls that we have in place. There is great sources of information on the beetle itself and on the control measures with the landing page on the .gov website. Alan's already mentioned Tree Alert. The Forest Research Ips page is very detailed. The EPO page has got further information on distribution, and if you want to have a look for further information on bark beetles in general, there's some very good documents now coming out of particularly collaborative European groups, and this one's a particularly interesting one that's reflecting how continental Europe is living with bark beetle outbreaks of varying degrees. So that's it there. Please feel free to contact me directly for further information. If there is time for any questions, I'll hand back to Caroline, and thank you all for your attention.
That's lovely. Thank you, Nick. That was really thorough. I really appreciate that. Just got a couple of quick questions. The first one is for Alan. It's a question from Angela asking about ramorum. So concerning the distinctive red reactive sub-bark layer on larch, is that unique to trees that are infected, or can it occur in other circumstances?
Not that I have observed. I have observed that reactionary staining in a couple of other species as well, but they were sort of weird ornamental bits and pieces in heritage gardens down in the southwest. So no, not that I've observed, but that doesn't mean it's not possible.
Okay, lovely. And then a couple of questions for Nick. So there's a two-part question here from Bradley. Do we need to put more emphasis on growing our own tree stock in the UK to reduce the amount of tree stock imported? I'll throw that over to both of you if you want.
That's a very wide question. We know that live plants are important pathways for pests and diseases. And there are some well-documented outbreaks and infestations with some insects and some pests. There's really we can apply some control with restrictions on what products come in, but in some cases it's a very difficult thing because weather and climatic conditions are bringing these pests to our shores anyway, and with particular reference to Ips typographus, the trade in live plants won't be relevant because live plants of that size for establishment purposes are too small to host the beetle. And through our intensified trapping and surveillance program over the last couple of years, we are aware that there are periodic beetles in effect landing in GB that we're picking up with our pheromone traps that are they're just flying across the channel. The proximity to the pests and diseases in the southeast is particularly difficult. It's a short hop across the channel for any windborne pest or pathogen. The climate in southeast England is very much more conducive than one in other parts of the country, and is only going to be getting more conducive to a lot of these beetles, which have often been kept at bay just because of the conditions we have in GB, historic conditions, and a lot of the pests and pathogens we're seeing are now sort of on the extent of their climatic range. So greater controls on material that comes in, including plant species, one thing, and greater control and understanding of pests and pathways. A lot of my work is focused at the border, where we're looking at wood and wood packaging that can host some of these things. What we don't know often is exactly what's being traded, and the rate of trade is very much more rapid nowadays, which is brought into it. So without going into all the detail, I would say yes in part, but it's not by no means the whole story, and is possibly in terms of IPS not the most important control. I hope that answers your question. If you want to take that offline with me, I've got some supporting data that I can provide on that sort of question as well.
Brilliant. Thank you, Nick. So the second part of Brad's question is, and you may feel the need to plead the fifth on this one, but do you, Nick, feel you have a large enough team and resources to tackle the pests and diseases entering the country from imports?
Really good question. I'm often asked that. I will probably always say we could do with more, and I think we could do with more. And we've seen an increase in numbers at the border for both ourselves and other organisations in recent years, and the whole notion of pest management and the risk through or the impact of pests and diseases on woodland is a hugely more important position within government thinking now than it was 10 years ago. The Chief Plant Health Officer a few years ago, when asked what keeps her awake at night, said pest and pathogens of trees and woodland, not pests and pathogens of potatoes or animals or grain, but the recognition that the impact of pests and diseases have on woodland are so far-reaching in terms of landscape, nature conservation, well-being, biodiversity, all these things is very well understood at government level, and is being resourced in that respect. Saying that, we can always do with more people. I think we can do with more resources in other ways, and there's lots of work going on with technological improvements, getting data about what materials are happening. There's a very important role for horizon scanning, collaboration with other plant health organisations around the globe. Trade has a part to play in terms of engagement with us in terms of where material is coming from. So the answer is we could always do with more, but it's not just a question of bodies or people. It's about information, it's about technology, it's about joined up working. It's about, I think, a general wider understanding across the population and trade about the risks, and getting buy-in from everyone to play their part.
Okay, fabulous. Final couple of quick questions from Andrew. Regarding IPS, you mentioned infections in Europe. Does this include the Republic of Ireland?
Okay, no. Republic of Ireland has actually not got any IPS infestation there, and they've actually got another level of protection over that we have in GB, in that we have a couple of pests that are now present in either some, most, or all of GB, including Dendroctons micans, which are currently not found in Ireland. So actually, Ireland has another level of protection against us. It's generally recognised to be slightly safer to be bringing material in from Ireland into GB than transporting it the other way, and there are close controls on that trade, and there's a long-standing programme of inspections of any material heading from GB, particularly the pest-free area in Scotland, to both Northern Ireland and Southern Ireland.
Okay. Final question is, what climatic conditions does IPS require in terms of mean summer temperatures and rainfall?
Well, in terms of breeding, I think it really needs to sort of get above 15, and I will again the detail of that is available through the forest research pages. I think the important point, or the important factor in terms of our infestation, is that there were very large numbers that developed on the continent, which coincided with 2 long hot summers where we had not only very hot high temperatures, but we had an extended period of relatively warm and sunny weather. If it seems a long time ago now, but the summer of 2018 started early and then it continued, and that allowed what was possibly a very small population in a very, very isolated part of one woodland to have 1 or 2 breeding cycles which suddenly used up all their host material, and it's at that point that they start looking around and start moving for and spreading outwards. So it's not and the timing of that was coinciding with particular weather patterns where we were getting extended periods of prevailing weather coming over from the continent. So I think climatically, southeast England is the sort of range of the natural opportunity for this beetle, but we don't quite know how it will respond. We don't know quite how the climate's changing. So it's in a way it's a useful beacon or sentinel. It's the southeast of England, but we're looking at a lot of work now to try and track whether individual or lone beetles are moving one further west towards our plantations and the more extended areas of spruce, and there's work going on in terms of whether they'd actually be able to survive in those conditions. High rainfall is not good for IPS because it disrupts their movement and breeding. On the other hand, very waterlogged and stressed stands of spruce is favourable for IPS establishment. So I won't quote any explicit numbers, but longer, warmer summers are attractive to the beetles.
Brilliant. We've no more questions, so I just want to say a really big thank you to Nick and Alan for presenting for us today. Thank you to everyone who's attended. It's been great to have you here. Yeah, reminder, there's still spaces on our other courses for the rest of the week, so you're able to book onto those until 10 AM on the day. So that's OPM tomorrow, plant passporting and biosecurity on Thursday, and deer on Friday. Just a quick highlight as well as the workshop that Forest Research are running this evening that you've got the email address for on screen, or you should do. I just wanted also to highlight that the Forestry Commission are really interested in talking to anyone that's got spruce in Kent and East Sussex to drop us an email so we can talk about how you can best manage your site to help prevent IPS. So if you want to drop me an email, my email address is caroline.gooch@forestrycommission.gov.uk. So yeah, so once again, thank you everyone for coming. I hope you've found this a really good experience, and we will be sending out the questionnaire on Friday. So thank you everyone.
Thank you, Caroline. Thanks for giving us the opportunity.